Welcome to the Planet MicroCap Showcase Virtual 2022. I'm your host, Robert Kraft, and now presenting at our event is Greg Duncan, Chairman and CEO of Virios Therapeutics. It's a publicly traded company. The symbol is VIRI on Nasdaq. Greg, take it away.
Thank you, Robert, and thank you to all of the attendees at the Planet MicroCap conference who have an interest in learning more about Virios Therapeutics. Virios Therapeutics is a development stage biotechnology company exploring the connection between activated herpes virus and its role as a catalyst in chronic diseases like fibromyalgia, irritable bowel syndrome, and we think possibly even long COVID. And the focus of the company is to take those activated viruses back into a dormant state, allow the immune system to reset, and to deliver clinical benefits for patients with these chronic disabling disorders. It may surprise you to know that estimates range from 320,000 to several trillions of viruses infect each and every human walking the planet known as Earth. And as troubling as that may sound, the good news is your immune system is healthy enough to control those viruses, to take them into a dormant state, and to keep them there for the vast majority of your lifetime. But for diseases like fibromyalgia, in our view, once that virus gets activated, it leads to the disease sequelae that is so disabling to patients with these particular disorders. Virios is a publicly traded company. As Robert mentioned, we have a tremendous amount of information available to you if you can visit EDGAR or the SEC website. I'll start by talking a little bit about the Virios Therapeutics team. This is a team with a strong track record of both drug development and commercialization. We have worked at small, mid-sized, and the largest pharmaceutical companies like Pfizer, for example. We have worked on both the public and the private side, and we have developed a number of drugs that are household names across the globe. For example, my first drug launch was a drug called Zoloft. Importantly, between myself and our board of directors, we've been directly responsible for either the development and or the commercialization of 2 of the 3 FDA-approved medicines to treat fibromyalgia, those being Lyrica and Savella. So we've got a team with a very deep track record in general, but very specific expertise here in the fibromyalgia development vertical. A little bit about the disease known as fibromyalgia. This is a disease that impacts 2 to 4% of the population. The hallmark characteristics of fibromyalgia are chronic widespread pain and very severe fatigue. When these symptoms are persistent for 3 or more months and we rule out other causes of the pain, we land in a fibromyalgia diagnosis. The challenge with making an accurate diagnosis is that the pain and fatigue is often comorbid with GI issues, headache, anxiety, depression, and sleep challenges. And so as a consequence, it can take patients months, if not years, to get to an accurate diagnosis. This is a disease with a higher prevalence in females and males. In fact, 70% of the population with fibromyalgia are female by gender, and 90% of the patients treated for fibromyalgia are female. There's devastating impacts of this particular disease, including a 3 times greater risk of committing suicide. These patients are high consumers of healthcare resources, and they have significant disability. Importantly, estimates range across the board, but we see as many as 40% of fibromyalgia patients being treated with opioids. And as I referenced earlier, there's only 3 FDA-approved medicines. That, unfortunately, has led to very significant dissatisfaction in the fibromyalgia patient community and amongst doctors who treat these patients. The key issue driving that dissatisfaction is poor tolerability of the 3 approved medications. Lyrica, Savella, and Cymbalta, those 3 approved medicines from FDA, are all CNS-mediated in their mechanistic approach. As a consequence, they come with downstream tolerability issues. So there's a very significant opportunity in this already large marketplace to develop a safe, tolerable, and effective new treatment that we believe could become a category leader. For context, this is a global marketplace dominated by North America and more specifically the US, with about 53% of sales. The top utilized drug in the space by sales is actually Lyrica, with sales somewhere between 1.5 and 2 billion. Savella and Cymbalta represent another $1.5 billion to 2 billion in sales in total. So as a consequence, this is a several billion-dollar market today before the next drug comes along. And using compounded annual growth rate from a variety of estimates, we expect the marketplace to be around $2.5 billion by 2030. A little bit about our hypothesis and why we're developing combination antiviral therapies to treat these patients. We believe that herpes viruses are the bad actors leading to the flare-ups, if you will, of these symptoms. We believe the latent herpes virus is actually infecting the neural ganglia or central nervous system in patients with fibromyalgia. Something stresses this virus and converts it from a dormant state to an active state. Once stressed, that virus creates more copies of itself using its own DNA. Specifically, DNA polymerase is used by the virus to create more copies of these particular cells. What is less well known, but germane to our thesis, is that upregulation of a common prostaglandin, cyclooxygenase 2, or COX-2 as I'll refer to it, is used by the virus to accelerate its replication. Think of COX-2 as a turbo booster for a sports car, if you will. You hit the gas, you accelerate. In the presence of COX-2, we get that significant upgrade or acceleration of the speed with which the virus creates more copies of itself. We believe this HSV1 activation specifically triggers an abnormal immune response and the pain, fatigue, and other symptoms that I referenced earlier that are the hallmark of this particular disease. Our approach is elegant and simple. We combine either famciclovir or valciclovir, which are two drugs that specifically inhibit viral DNA polymerase. These are the materials used by the virus to create more copies of itself. This attacks the DNA specifically. And we combine that with celecoxib, which is a COX-2 inhibitor. What the COX-2 inhibitor does is it binds to that COX-2 pocket, thereby leaving less COX-2 available to be consumed by the virus to accelerate its replication. We attack the virus, we turn off the turbo booster, that inhibits viral replication. That takes the active virus back into a dormant state and allows the immune system to reset. And it is our belief that either the resetting of the immune system and or the virus going back into a dormant state is what's leading to the clinical benefits we've seen in trials to date with our lead compound, IMC-1. This has been a very purposeful research program. The founder of the company, Dr. Skip Pridgeon, William Pridgeon more formally, conducted a GI biopsy study where he identified active replicating virus in the GI tissue of patients with fibromyalgia and a functional GI disorder. Highly statistically significant upregulation of active replicating infection in the gut tissue of these patients. That led to our phase 2A clinical program, where we showed very clearly that IMC-1, dosed twice daily, this is the combination of famciclovir and valciclovir into a fixed dose combination, showed statistically significant reductions in pain at the 16-week pre-specified endpoint. This is the primary endpoint that FDA has used to approve the prior 3 medications. So we have tie-in of the mechanism, we have clinical data showing effectiveness of our lead compound on the endpoint that FDA cares about for approval over time. That led us to our phase 2B program. The phase 2B program is known as the Fortress program. And specifically, we enrolled 425 female patients aged 18 to 65. And we compared IMC-1 at doses higher than the previous phase 2A study, specifically 675 milligrams of famciclovir plus 180 milligrams of celecoxib in a fixed dose capsule, and compared that with placebo, both dosed twice daily. This was a study conducted here in the US with over 40 sites, and we used the standard diagnostic criteria from the American College of Rheumatology. The primary endpoint in this trial was reduction in pain, the very same endpoint we used in the previously successful phase 2A trial. We looked at all of the key secondary endpoints that FDA cares about, including global improvement, specific domains of the disease, and different pain responder threshold analyses. In this 16-week trial, patients were dosed to either IMC-1 or placebo and treated for 14 weeks. At week 14, we converted all patients to placebo. Those on placebo stayed on placebo, and those on IMC-1 were converted to placebo to see what the washout of IMC-1 would look like. But specifically, we prospectively defined week 14 as the primary endpoint analysis for the statistics to determine the effectiveness of IMC-1. From a disposition perspective, IMC-1 performed extraordinarily well. We had a higher completion rate overall on IMC-1 than placebo, and we had a lower discontinuation rate due to side effects on IMC-1 versus even placebo. In fact, about half the rate of dropout due to adverse events on IMC-1 versus placebo. This is very unusual. As I referenced before, the 3 approved medicines come with significant side effects. You can see side effects in the range of, in the low teens to upwards of 30 or 35%, depending on which drug you look at. In our particular trial here, 216 patients with IMC1 showed a dropout rate of 4.6%. That compares very favorably, albeit not in a head-to-head trial, to the double-digit dropout rate in Lyrica and dropout rates for Cymbalta and Savella that are north of 20%. So we see a very well-tolerated profile for IMC1. Specifically, if we take a look at how IMC1 compares to those other three approved medicines, it may surprise you to know that the very largest side effect we had in the trial, at about 8% for IMC1, was actually COVID-19, patients getting infected with COVID-19 while they're in the midst of our trial. Every other side effect that led to discontinuation for IMC1 was below 5%. Look how that compares to the roughly 40% dizziness on Lyrica, or the 37% nausea on Savella, or the 23% nausea on Cymbalta. This is a drug that looks to be very well tolerated, and that could be a very key distinguishing feature for IMC1 moving forward. Unfortunately, the primary endpoint did not meet statistical significance at the pre-specified week 14 endpoint. This was very disappointing to everybody, including me, who's a shareholder in Virios Therapeutics. And we decided to explore more in the data, really, frankly, based on the recommendation of our statistical consultants, who had conveyed to us they had seen in several cases, both fibromyalgia cases, where we're looking at pain, and general pain studies, that they had obscure, if you will, bifurcation of events for trials that were conducted running up and into COVID, and for trials that were run during the COVID pandemic. And so we decided to take a deeper look. And in fact, we're very pleased to see, intriguingly, that of the 50% of patients that were enrolled in the first half of the trial, we'll call these patients the cohort 1, we showed an effect of IMC1, but unfortunately we showed a placebo response that was greater than anything we've ever seen on drug or placebo in the past. And as a consequence, roughly half of the patients in the first half of the trial did not show any statistical significance, even though the drug worked quite well. The placebo response was way beyond anything we've ever forecasted or seen in prior trials. Cohort 2, on the other hand, showed highly statistically significant reduction in pain when we compare IMC1 versus placebo. This is the second 50% of patients that were enrolled in the trial. So we decided to dig into this analysis because we don't think this is due to chance. If we didn't change the endpoint, the treatment regimen, the time points for assessing patients, the types of patients, there is really no statistical reason for this unless there's something going on that's unique to these two cohorts. So we decided to explore that in greater detail. We looked at cohort 1 versus cohort 2, which I just shared with you. We looked at new patients versus patients that were prior fibromyalgia trial participants. We looked at a whole host of different things like length of treatment, length of diagnosis, and I'm pleased to share with you we have found what we think is a very actionable way to continue the development of IMC1 as a viable treatment option for patients with fibromyalgia. Let me share a little bit more of those analyses. The first impact I referenced was this cohort 1, cohort 2. Cohort 1 was recruited between June 2021 and November 3rd of 2021. This may be hard to remember because we've come a long way since COVID really changed all of our lives, but this is when the Delta variant of COVID was most dominant. We had very significant quarantining. In fact, we had patients going from quarantining in their house to the study sites and back. Remember quarantine, it was not a very fun experience. We had research site staffing issues at a number of sites across the country. And in fact, 70% of the patients that were recruited early on in this trial were either prior fibromyalgia trial participants or had prior site engagement. Specifically, their names were on the list of the local site as being part of their overall care, either through research or community care. Contrast that with cohort 2, where the quarantine was actually lifted in the vast majority of geographies. Research sites staffing stabilized. And in this particular back end of the trial, we actually had the vast majority of patients recruited through advertising. These were patients that were new to the site, and in most cases, new to fibromyalgia research. So we do think, while we can't specifically identify one key issue, we do think the tentacles, as I like to call it, of COVID had an impact on that placebo response in the first half of the trial. And I've shared with you, unfortunately, it was a very robust response. In fact, a response on placebo greater than drug, which was in stark contrast to the cohort 2 patients when we got back to a more normalized timeframe. This is when the Omicron variant became the dominant virus here in the US. The second thing we found was that patients that were new to fibromyalgia research. These are patients recruited primarily through advertising. They were new to the site, they were new to fibromyalgia research, showed highly statistically significant reductions in pain at a p-value of 0.016. Compare that to the existing patients. These are patients that were in prior trials or previously treated or known to the site. We believe, based on our analysis here, and we vetted this and gotten corroboration from key opinion leaders in this space, that the existing patient group in fibromyalgia may be becoming more refractory. We've seen this in other categories like depression, where we just researched so much, there were so many programs, and we can tell you there's probably 6 to 8 programs over the last 6 to 8 years just in the fibromyalgia vertical. Those patients that go from trial to trial to trial may be becoming more resistant to treatment. So very key finding here is that patients who are new to the research paradigm, if you will, more naive to treatment, showed a highly robust response to IMC-1 treatment. That's something we can action moving forward as we move into continued development with IMC-1. For example, if you look at those new patients over the course of the entire trial, so this effect was irrespective of which COVID variant was dominant in the US, we see statistical significant reduction in pain at a p-value of 0.016%. What I'd also highlight is the last approved drug, which was Savella. If you look at the curves of the drug, which are in the orange and blue, and compare that to placebo, you can see at this time when Savella was assessed in its own research program, when the entire community was more naive, or basically everybody was new to clinical research, the data are quite parallel. And so it is key for us to understand that new patients responded well to IMC-1, irrespective of when they enrolled in the trial. It's a very key finding for us, and those data look quite similar to the last approved fibromyalgia treatment, that being Savella. That effect was not just seen on pain. That effect was seen in a reduction in fatigue, improvement in overall fibromyalgia symptoms, and in patients' global impression of their overall health status at p-values of between 0.001 and 0.005. In addition, we saw a reduction in depression symptoms and anxiety symptoms. Recall, I made a statement at the early part of today's presentation where we think this virus is the bad actor that's triggering fibromyalgia flare-ups and symptoms. We think this is a root cause of the disease. And you can see, based on all the data I'm just sharing with you, be it pain, fatigue, overall health, depression, and anxiety, by getting this virus back into a dormant state, we're having a very profound impact for patients who are new to research trials in fibromyalgia. The other thing we noticed was that research sites that were specifically executing and conducting research versus those that were a combination of research and community care did quite a bit better. Specifically, the sites that were specifically doing just research, not providing community care in their local geographies, performed quite well. Their placebo response rate was more normal. They did not have as many staffing issues. And so the second key factor we found here, in addition to timing and new patients, is that conducting research with purely genuine research sites, as opposed to these mixed research and community care sites, also delivered highly statistically significant effects of drug versus placebo. So that will be one of the things we manage moving forward. If we were to define our optimal patient, we'd be looking at the second half of the pandemic, and we'd be looking at recruiting just new patients. So how would that synthesis, that diagnostic that's a little bit more refined compared to the Fortress study, bear out? Well, you can see the results. We had 104 patients that were recruited that were new to fibromyalgia research in the second half of the trial. And you can see a very, very robust p-value of 0.021 on their pain scores. This is a sample size about 1/4 of what we thought we needed to reflect IMC-1's potential. And so we're very excited about moving forward, focused on new patients in a more normalized environment, which is what we see moving forward. You can also see on the right-hand side of slide number 21 that when the drug is washed out, weeks 15 and 16, patients' pain scores begin to approach that of placebo. We're washing out the drug, which we think is a good harbinger of the effectiveness of IMC-1. So overall, while we were disappointed that IMC-1 treatment did not achieve statistically significant pain reduction, we do believe the pandemic effect had a major impact on this very robust, unfortunate placebo response. That could be related to quarantining, leaving quarantine, going to the site and coming back. Just that interaction can drive a placebo response. We think site staff disruption and turnover was also another big impact here, because the people we trained on how to effectively assess patients, in many cases, vacated the sites where they were originally employed. The treatment effect differences over time are striking, such that we do not believe this is a random effect. I would also mention that, very importantly, IMC-1 safety profile was even better than we anticipated and better than prior studies. And we believe that focusing on new or naive treatment patients to treat with IMC-1, recruited via advertising, in many cases, can deliver a much more robust outcome. And we believe we can demonstrate IMC-1's potential in this specific group by screening out patients who've been involved in prior research, who may, in many cases, be becoming more refractory. So that's the synthesis of the Fortress trial. We do have another ongoing program in long COVID. I won't go into too much detail here because this is an exploratory program, but we're assessing a combination of famciclovir and celecoxib, so a different antiviral plus celecoxib in this case, to treat the symptoms that are associated with long COVID sequelae, which I'm sure all of you are familiar with right now. Unfortunately, some of you may be suffering from it or have suffered from it in the past. The most persistent symptoms of long COVID sequelae are fatigue, difficulty breathing, memory loss or brain fog, and problems with attention. We do know that the COVID-19 infection disrupts immune systems. It has a direct effect on the neurons responsible for your immune response, and it creates psychological stress and fever. Herpes virus reactivation, we believe, is the problem here. We believe this is stacking COVID on top of already dominant herpes and other viruses, is stressing out the immune system. It's triggering reactivation of those latent herpes viruses. And we believe neurocognitive impairment and fatigue are the bad actors here, resulting from Epstein-Barr reactivation. You're probably familiar with a virus known as Epstein-Barr. This is a herpes virus, just a specific form of herpes virus. And we believe it is the activation of that previously dormant Epstein-Barr, which is triggering the fatigue, the attention disorder, the memory loss that is a hallmark of these patients. We will be recruiting these patients into this trial, this exploratory trial, through probably mid to late January, and we would expect results sometime, hopefully, in the April-May time period, roughly Q2, although timing is dependent upon the site. This is an unrestricted investigational grant to the Bateman Horne Center. They are conducting the study on our behalf. So to summarize, we're really excited about what we found in the IMC-1 Fortress data, specifically focusing on patients that are new to research, more naive patients that have not been involved in previous trials, and conducting the program specifically with centers that focus on nothing but clinical research. We will be requesting an FDA meeting very shortly, sometime in the latter part of November or the very first week in December. In the first half of next year, we would expect FDA feedback on our proposed program. We will be proposing moving to phase 3 because of the safety data we've generated now in the Fortress trial. We believe support moving into the larger phase 3 programs. We will be assessing different forms of partnership or financing, whatever makes the most sense for our shareholders and ultimately for the success of IMC-1 as we move into the first half of 2023. And as I referenced earlier, we would expect the results from our exploratory long COVID program sometime in the first half of 2023. Moving forward, presuming alignment with FDA on our proposal to move to phase 3, we presume starting the phase 3 program in the early part of the second half of next year. With that, I want to thank you for your time and attention. And Robert, I'd be happy to answer any questions you may have.
Very good, Greg. Thank you so much for your presentation here today. My first question for you, and this is what I'm asking to everyone here presenting at the Planet MicroCap Showcase. You're managing through tough markets right now. What measures is the company taking to manage macroeconomic pressures that are currently affecting most microcap companies across the board?
Yeah, so the good news is shortly after the unblinding of the Fortress data, we actually raised capital, which may seem counterintuitive, but we guided people prior to the study results that we would need to raise more capital. So we actually have capital now, although we didn't like the valuation at the time. That gets us through the end of 2023. So it gives us the time to get down to FDA, figure out exactly what the next steps of IMC-1 research looks like. We believe the data support progressing to phase 3. And that also gives us the time to explore different forms of financing and or a combination of partnership and financing, depending on different regions of the world, for example, and a combination of that partnership and financing to progress IMC-1's development. So we have a bit of time to figure that out, get the nod from FDA, which we think will be good news for shareholders in Virios Therapeutics, and then decide which one of those paths makes the most sense going forward, because this is a very, very large market opportunity. I had Lyrica in my stable of products during my tenure at Pfizer. We know that Lyrica sales just for fibro were north of 1.5 billion. Cymbalta was in that range. Savella was a couple $100 million. So there's a established marketplace that is desperate for new therapies, and we believe well-tolerated new therapies, and we believe IMC-1 has that potential to be that category leader.
Absolutely. So let's look at the competitive landscape right now, because I would assume, maybe I'm wrong, but I would assume that there are other companies in clinical development that are also looking to just develop newer therapies, potentially better therapies than what's currently standard of care right now. So what does that competitive landscape look like? And are there others that are closer, or where are they at in the clinic?
Yeah, so right now there's 2 therapies that are out in development. Ours, IMC-1, and Tonix's drug, cyclobenzaprine, which is a drug that improves sleep, and by improving sleep has shown to be somewhat effective in reducing fibromyalgia symptoms. The Tonix team is actually in phase 3 right now. IMC-1 hopes to go to phase 3. I would reference that we're attacking 2 completely different mechanisms. So in some regards, these could be complementary therapies. I will tell you, it's looking at some of the Tonix experience and that of other programs in the space, because there's been about a half a dozen programs. I think 8 was our last count over the last decade, specific programs of drugs or different types of therapy. And we think our learnings are actually consistent with theirs, where these bifurcation of results is causing us to look in greater detail. And I think everybody's frankly suffering from having the same group of patients. And I mean no disrespect, they are desperate for new therapies, but from one trial to the next trial to the next trial, they may be more refractory and not necessarily representing the broader universe of fibromyalgia patients. And so I think you'll see 2 drugs moving forward that actually could be used in a very complementary fashion. Unfortunately, based on the history of those last few programs, there's very few other programs in the space at present. Aptinix just had a bad result, as did a few other companies. So it's really back down to Virios Therapeutics and Tonix.
Very good. All right. So final question for you here today, and this is more of a wrap question because you kind of went over it in your last couple slides there. You know, from what you can tell us, looking ahead now at 2023, what would you say are some of the company's value catalysts that you think folks that are listening or watching right now should pay attention to?
Sure. So I think we're all hoping that this dismal marketplace we're all suffering in eventually evolves and things settle down on the geopolitical front and all other related fronts. And we do have capital to get us through this, the next 12 months, 14 months anyway. But I think the next key catalysts for us will be, 1, FDA alignment on what the fibromyalgia program will look like. We expect that sometime in the first half of next year. I can't guarantee you when that meeting will be because we're requesting an in-person meeting. And we do know that the FDA dockets are quite full, so that could be March, April. Could come forward or back a little bit, but we hope to have FDA feedback in the first half of next year. The other exciting program, which we don't talk a lot about, which could have as much value as a fibromyalgia program, is a long COVID program. We should announce full recruitment of that program sometime in the early part of Q1, and we'll have results roughly 12 weeks after that. So maybe call it 14 weeks after that, sometime towards the back half of Q2 or Q3, we'll actually unveil the long COVID data. That could be a very exciting program because I think, Robert, you'd probably agree, each and every one of us has now touched it either personally or have somebody, family or friend who's actually suffered from the long-term consequences of COVID. And I see people all the time that, their fatigue still there. They can't focus. And so that's a second key catalyst in the first half of 2023. That will give us, hopefully, the valuation bump to allow us to either go raise more money and or sign up some form of partnership plus capital scaling to progress into the next phase of research, which we hope will be a full-blown phase 3 program.
Absolutely. Well, Greg, thank you so much for your presentation today. We really do appreciate it. And to everybody watching and listening in and engaging with this, thank you for watching today.
Thank you, Robert. We appreciate it.